Yeah. So here we just got stopped here where the leading strand, you can see the polymerase enzyme is helping to lead the strand by formation of a new daughter strand. Whereas the other strand, which is lacking, there are certain obstacles since it is running from 3 prime to 5 prime. So it has a problem. And the other strand, the lagging strand, cannot be made it in a continuous way. So that's the reason here there is formation of the Okazaki fragments. Let us see what happens. So since it runs in an opposite direction, the DNA polymerase can therefore only make this strand in a series of small chunks. So you can see there are small chunks, small pockets of DNA form. So these fragments are called as Okazaki. Since they were discovered by the Okazaki scientist, the fragments were named after his discovery. So the DNA, yes. Any doubt? Shall I proceed? Yeah. So the DNA polymerase can so as we know, in the lagging strand, since it lags, so the DNA polymerase can therefore only make this strand in a series of small chunks. So small, as you can see, these are the RNA primers. And after this, you will be getting small chunks of DNA. Okay. So this blue color is the RNA primer, which will be initiating the strand to form the new DNA. And these small chunks are called as the Okazaki fragments. So as you can see here, these yeah, as I mentioned, these are the Okazaki fragments. And later on, these Okazaki fragments are joined. So you can see that each fragment is started with RNA primer. The dark blue are the RNA primers and the new chunks of DNA, which the DNA polymerase joins from 5 prime to 3 prime directions are the Okazaki fragments. I think this video will give you a clear picture how the lagging strand has been giving rise to the daughter strand. Okay, so there are n number of Okazaki fragments which are made and process repeated again and again since a new DNA has made. Then this particular exonuclease, the enzyme exonuclease removes all the DNA primers from both the strands of DNA. That is the leading strand as well as the lagging strands. Excision. You can see the excision, the removal of these particular primers is being taken up by the exonuclease enzyme. And after it removes, you can see that your another DNA polymerase enzyme is coming up and joining, and this is filling up the gaps which are being formed by the removal of the RNA primers. So another DNA polymerase enzyme will fill the gaps that are left behind by the removal of the RNA primers by exonuclease enzyme. So after they are formed with the help of the ligase, okay, next comes the ligase enzyme. This ligase helps in sealing the fragments of the DNA, wherever the gaps, the attachments, you can see the attachment here, right? So this ligase enzyme helps in attaching the daughter DNA. Okay, so yeah, so in both the strands, you can see the double stranded DNA form. So that's how the replication takes place, and the DNA replication is described as semi-conservative replication. And you can see each DNA molecule is made up of one old one, and that is having the information, that is having the parental message which has been conserved in that particular strand. So this video I've taken from YouTube. Whenever you have any doubts, you can just go through. There are many animated pictures of the DNA replication. So you can just check it out in the YouTube. So as we have seen that, now let us go in depth of it, how this is occurring. So you can see that this DNA, yeah. So the replication generally takes place in the S phase of the cell multiplication, like in mitosis, the cell cycle. In the mitosis, you have your G1 phase, S phase, and G2 phase, right? So this replication takes place in the S phase of the mitosis division. So as you can see that, so the ori region, you can see in the ori region, the replication begins, the two strands open up forming the replication, and you can see the Y-shaped region, that is the fork, and the new strands grows at the fork, and you can see this particular direction of the replication fork. Excuse me. You can see the DNA strands open up, and this, it's a continuous procedure where the DNA open up and you can see the replication bubble formation. And these replication bubbles, you can see in prokaryotes single bubble, and in eukaryotes you can see many bubbles. So this one is representing the eukaryote, I can say. And as we mentioned, the helicase will be unwinding and separates the two strands of DNA by breaking the weak hydrogen bonds. And you can see the single strand binding proteins attach and keep the two DNA strands separated and untwisted. So here you can see the topoisomerase enzyme, which has been attached to two forks of the bubble to relieve the stress on the DNA molecule as it gets separated. So as you can see, the RNA primers are present in the start addition of the new nucleotides, and the primase enzyme that synthesize the RNA primer, and the DNA polymerase can then add the new nucleotides. So the primase helps in RNA primer, and the DNA polymerase helps in the new nucleotides to attach to the DNA fork. So as you can see, this helicase is separating the DNA. Okay, and the RNA primer is being attached with the help of the primase enzyme. This will help in the synthesis of new strand. So both the leading as well as the lagging strand has RNA primers which are attached by the primase enzyme at the origin of replication. So that's how the DNA replication starts, and you can see that this DNA polymerase enzyme starts adding your nucleotides. Okay, so as the nucleotides are added to the 3' end of the DNA, and this causes the new strand to build in 5' to 3' direction. So you can see this 5' to 3' is your parental strand, to which the DNA polymerase is adding the nucleotides, and you can see again from 5' to 3' a new strand is being formed in one direction and in the other direction also. So the direction of replication, you can see that this DNA polymerase is adding the nucleotide to the 3' end. Okay, and the new strand is forming from 5' to 3' end. And now you can see how it remembers the carbons are numbered. We have already seen how it is 3' to 5'. So 5' at the 5 carbon end, the phosphate group is ended. This is your 5', right? And in the 3', if it is at the carbon group 3. So here comes. So the remember strands are anti-parallel to each other. You can see the 5'. Okay, where it is ending up with the attachment at the 3'. So the 3 goes and 5 at the 3 carbon goes and attached to the 5 carbon, and so on. And you can see that ATGC, the nitrogenous base pairing. So the bases always pair up as ATGC. Now the synthesis of new strands. The leading strand is synthesized as a single strand from point of origin towards opening replication fork, which we have already discussed. And here comes your leading strand. In this leading strand, you're seeing the addition of your nucleotides without any hazels by the DNA polymerase, since it is the leading strand, whereas in the lagging strand, it is synthesized discontinuously against overall direction of the replication. And this strand is made in many short segments, and it is replicated from replication fork towards the origin. That's why it is bidirectional. And as you can see here, your RNA primer, a single RNA primer is there in the leading strand, whereas you can see multiple RNA primers on the lagging strand. So as you can see, the lagging strand, the Okazaki fragment, series of short segments on the lagging strands you can see here. This is the primer. Okay, yeah, sorry, the light blue color is your primer. And to this, the Okazaki fragments are synthesized. And this Okazaki fragments, again, they are stapled and attached to form the new strand. And these Okazaki, as I mentioned, they're stapled or joined with the help of the ligase enzyme. So the DNA ligase helps in joining of the Okazaki fragment. Now you can see the replication fork and the point of origin, and you can see how the fork has been maintained in between the lagging and the leading strand. So you can see the proofreading is done by, we already mentioned, the DNA polymerase enzyme helps in proofreading. It initially makes about 1 in 10,000 base pairing errors, and you can see the enzyme proofreads and corrects these mistakes by excision, and the new error rate for DNA that has been proofread is 1 in 1 billion base pairing errors. So the error occurring is 1 in 1 billion. So semi-conservative model of replication is an ideal one represented, and it is a idea presented by Watson and Crick. And the two strands of the parental molecule separates each act as a template for new complement strand, and you can see that new DNA conserve one parental origin, which is being a repeat of the semi-conservative. And you can see in the semi-conservative, the parental is being conserved and the dispersive in between. In conservative, there is a new strand formed. So this we have already seen, and here comes the DNA damage and the repair, where the chemicals and ultraviolet radiation damage the DNA in our body cells. The cells must continuously repair the DNA damage. If not, it leads to mutation and leads to genotypic and phenotypic characters which are mismatching with the original one. And this excision repair occurs when any of over 50 repair enzymes remove the damaged part of the DNA, and the DNA polymerase and DNA ligase replace and bond the new nucleotide together. So as you can see, there's a small question. What would be the complementary DNA strand for the following DNA sequence, that is DNA running from 5' to 3', that is CGTATG. So I think it's a very simple one. What it will be with C? Is it A or is it T? Or it is G? So you can see the answer will be G, C, A, T, A, C. Right? G, C, A, T, A, C. So the post-replicative modification of DNA, you have the methylation, where one of the major post-replication reaction. And this is where the site of methylation of eukaryotic, where the DNA is like this is post-replicated modification occurs after the termination of the replication. As you can see, the DNA is always on cytosine residue in CG dinucleotide. And this DNA methylation plays an important role in epigenetic gene regulation in development of the disease. And these epigenetics is nothing but what is this epigenetics? It refers to the study of changes in the regulation of gene activity and expression that are not dependent on gene DNA sequence. And these epigenetics often refers to the study of single genes or sets of genes. And these epigenomics refers to the more global analysis of epigenetic changes across the entire genome. So the changes which regulate the gene activity and expression, we refer it as epigenetics, which are not dependent on DNA sequence. So as you can see, the epigenetic mechanism, they refer to the RNA interference, where the DNA expression is histone modification and DNA methylation. This may bonds for you. This methylation we can go in depth when we study the DNA disorders, repair mechanism, excision mechanism. So I would like to end my session here. If any doubts, any queries, please message me. Or you can take my number, you can post me, and as well as you can take help of sir also, you can get back to me. Sir, next class I will take the structures of these enzymes involved in DNA replication, so that this will help us to understand how these enzymes help us for DNA replication, and we can come up with any models also. So this I could give you more picture on it, where we can also study in depth of the DNA repair mechanism, the DNA damage mechanism also, if we understand the structure of these particular enzymes. Hope my session was clear. Sir?
Okay, madam.
Yes, sir. So next class I will continue with the enzymes and all, sir.
Sure,
Am
madam.
I clear with this session? Yes, sir. Okay,
Yes,
sir.
madam.
Thank you, sir. Thank you
Right,
for giving me this opportunity.
Okay, thank you very much, madam. Clearly explained today's class, madam. So our participants, our students also very enlightened. So tomorrow, sharp 12 p.m., 12 o'clock, same time, madam is going to explain which type of enzymes are involved in the DNA replication process. Okay, one of the beautiful class tomorrow. My dear students, let's join sharp 12 p.m. tomorrow class. Thank you, madam.
Okay. Yeah. Thank you, sir.
Thank you very much.